Hello and welcome to my final presentation. For my scenario, I chose burglary. In this scenario, a house was broken into and burglarized by at least one unknown perpetrator. The house did show signs of a break-in, as a window at the back of the house was broken. Additionally, a small red-brown stain was found and collected with 2 sterile swabs. In the aftermath of the burglary, the victims reported that a knife they did not own was left behind on the kitchen counter. Its handle was swabbed for evidence. Lastly, an anonymous person called in a tip to the Edmond Police Department, which led to the arrest of John Smith and Michelle Jones. An electrophoregram is a chart produced after using electrophoresis. The electrophoregram was used on all DNA samples in this instance. It works by separating RNA and DNA with the help of their electric charge and size. With the help of our profiles, we can now put the necessary information into a table. I started the table by writing out the case number, the name of the analyst, the date, and page number at the top. Then, the names of the 2 swab samples in the top 2 boxes of the sample column, and the names for the reference samples of John Smith and Michelle Jones in the 2 bottom boxes. Next, I looked at the electrophorograms to locate the alleles so that I could fill in the table for each sample. I did so by matching the alleles under each locus to the corresponding locus column. For instance, say I want to fill in the D19 locus column on the table for John Smith. First, I would locate the D19 locus on said electrophorogram, which is right here. The section lasts for as long as the locus bar does, and underneath it is 1 box. The number at the top of the box is the allele. Next, I will plug it into the table. It is a 12. After comparing all the alleles to one another, I discovered one match. The alleles from reference sample 1, John Smith, match the alleles from evidence number 1, the blood on the window. Each allele from each genetic location matches each other. As far as CODIS eligibility goes, I believe John Smith's profile would be eligible for entry into the offender indices of CODIS. His is a complete, single-source profile that was compared to an item of forensic evidence. As far as other types of analyses goes, I will first show you how to differentiate between heterozygous and homozygous alleles. The data in this scenario has examples of both. We know that the allele under D8 in Michelle Jones' profile is homozygous because there's only 1 allele, yet there are 2 peaks, meaning there are 2 of the same allele. An example of a heterozygous allele would be found within D8 of John Smith's profile. There are 2 alleles, 12 and 14. Next up is the question of whether these profiles are single source or mixtures. John Smith's and Michelle Jones' profiles are both single source, as their DNA was presumably taken in the form of a buccal swab during booking for either this case or a past one. I am not so confident about the profile for the knife handle because there's additional information under locus TH01 and D16. I was leaning towards the possibility that it's a mixture of the DNA between both John Smith and Michelle Jones, but the alleles in the knife handle row do not seem to be consistent with a mixture of their profiles. It's likely that an unknown person's DNA was swapped from the knife handle, as deconvolution is often not possible with complex mixtures. Now, how do we know that the profiles are male or female? Well, John Smith's profile is male because there's an X and Y under the amelogenin locus. The same goes for the blood swab taken from the window as well as the swab from the knife handle. We can also see that Michelle Jones' profile reads as female because of the X. That is all for the presentation. Thank you so much for watching. I truly had a great time breaking down the profiles.